Good morning, class, and welcome to the last video going over Unit 6 exam. This one is a little bit of a less questions, so hopefully it'll go by quicker. After you finish with this exam on Teams, I will have a table, like a little file set up and little videos on there. If you know that you failed an EdSite, remember 60 or lower is failing, you can start watching some of those review videos, looking at your notes. The notes again are already posted in Teams. Also, if you are pending EdSites, again, do not use your notes, but you can go into EdSite and finish any of the quizzes. Remember, they're B.9A, B, and C, and B.5A, B, and C again. So there's a total of 6 EdSite quizzes that should have been completed. Some classes we've been running out of time and you only complete quiz 1 and 2. So again, with the time that we have left over for our biology block, finish your EdSites. That is the main goal. And after you finish your EdSites, or if you already finished them, you would watch those other support videos in case you failed a certain section. But again, let's get started and finish off our Unit 6 exam overview. So we're already on question 21. The diagram of DNA is provided. The arrow is pointing to which component of the DNA strand. So here, we see the different shapes. And that is what is in the middle of both of our DNAs. So that's what we're looking at here. The sugar deoxyribose and phosphate, remember that makes the backbone. It is not in the center. Genes, one from each parent. That is not a gene. The DNA is not showing us anything from mom or dad. Nitrogen bases held together by a hydrogen bond. Absolutely. So we see the bases here in the middle. That is the actual hydrogen bond. So that is a possible answer. The amino acids that will code for a protein. So again, it is not an amino acid that we see there, but we definitely see our bases. Remember, the structure is important. This line right here, this is your sugar phosphate backbone. What we see here, these shapes in the middle of both of them, these are nitrogen bases. Remember, we only have 4. Our 4 nitrogen bases are ATGC, adenine, thymine, guanine, and cytosine. And that is what makes up all life. Anything that is living, plants, animals, bacteria, ourselves, we are all made up of DNA. And that DNA has the 4 base structures, adenine, thymine, guanine, and cytosine. Moving on, which of these changes to the DNA triplet? So here we have, it does tell us it's in DNA GCT, will affect the protein produced. So we start off with GCT. We have to convert it into our messenger RNA. Remember, for mRNA, our bases are now AU, GC. So again, the G is with C, the C is with G, and the T is with A. CGA, so we have C, we have G, and we have A. Arginine is our amino acid. So now we're going to our answer choices and see which one is different. So GTT, again, is CAA, and the amino acid CAA is glutamine. So that one is going to affect the protein because it would be a different amino acid. Let's go on to the next one. TCT, that is AGA. So we have A, G, and A. That is also arginine. So that is like our original. This one is good. There is no change. We have TCC. So TCC is AGG. Let's go back to our chart. A, G, and G. It is arginine as well. So we should see here a little pattern. And then GCA is CGU. So we go back to our chart. C, G, and U is also arginine. So there is only one that would affect it, right? Which is A, because A is a different amino acid, which would affect the protein produced. A, because you would have glutamine instead of arginine. So again, this is a little bit more work. All you had to do was from your DNA make your messenger RNA, that is the transcription, and then do the translation part with your chart. But pretty easy overall. It's just knowing. The good thing is that this question did tell you DNA, so you know to read carefully, and that's how you would handle it the next time. So organisms can be classified based on homology, which is shared characteristics inherited from a common ancestor. In the past, homologies were based on studies of anatomical structures and patterns of embryonic development. In more recent years, the use of molecular biology techniques has allowed homologies to be compared at the nucleotide sequences. Nucleotide sequences comparisons are possible because all organisms share the following. So again, they give us a lot of information, but this is the meat of it. Our nucleotide sequences, all organisms share what? We all share, we know we share DNA, which is ATGC. So we do share DNA bases. We do not all share organelles. Remember, prokaryotes and eukaryotes have different ones. The division of nuclear chromosomes, not everyone goes through that specific process, and types of proteins needed for cellular functions. So remember, what do we all have in common? Our DNA bases. Our last unit is going to be homology, and homology is how we are all related. We are all related because we all have DNA, and DNA is made up of ATGC. So everything that is alive, again, humans, animals, plants, bacteria, anything that is considered living has DNA. So it is something that's a crazy concept that we all share one thing, even though we're all so different. So again, just process of elimination and prior knowledge should help us with this question. Ooh, here we have an image. We have transcription and we have translation occurring. So we know this is protein synthesis. Observe the illustration of the protein synthesis process. What statement correctly describes the process in the diagram above? So the growing protein has amino acids in the sequence coded by the cytoplasm. They're not really coded there. The two subunits of the ribosome translate DNA code into mRNA codons. The DNA is transcribed into ribosomes to perform proteins in the cytoplasm. Anticodon on the tRNA pair with the mRNA codons to form proteins. So again, we're just looking at the image. Here in translation, I think that's what it was talking about. So we're not growing the proteins coded, like the cytoplasm did not code anything. So process of elimination, this one's out. We are not translating to mRNA codons. Remember, in translation, you go from mRNA to proteins. Here it's telling us that you go from DNA to mRNA, that is transcription, so that is wrong. DNA is transcribed into ribosomes to form proteins in the cytoplasm. We do not form proteins in the cytoplasm. We form proteins in the ribosome, so this statement is incorrect. Anticodons in the tRNA, definitely. They match with mRNA codons to make amino acids. Yes. So this one, letter D is correct. It's specifically talking about this. So this is your tRNA. The anticodons match with the codons and translation is occurring. So again, if you see the image and you know the processes, which we did review multiple times, easy. Ooh, here we have the thorns on a bougainvillea. This question, I actually fixed it for you on the exam, guys. It wasn't something that we went over specifically. So I either gave you the credit or I told you what answer to choose before the unit exam. So again, the long, sharp spines of the bougainvillea plant develop in a cluster of flowers because the environment around the plant. So environmental factors affect cell differentiation. Remember, cell differentiation is caused by genes being turned on or off. So again, bougainvillea, a lot of us might know what that is. It is a plant that's really pretty, has really pretty flowers, but it also has spines. So again, depending on the environment, they are in hot valley environments. How does this affect the cell differentiation or what the cell specifically becomes? So let's start reading and use process of elimination. Altering the levels of regulatory molecules in the cell, acting randomly on genes that regulate cell differentiation. So randomly things are mutations. It is not a mutation. Remember, it is affected by the environment, causing practically all cells to stop transcription. It's not stopping anything. You're still making proteins and changing the base sequences of mRNA molecules. Again, when you change the base sequence, it is telling you that this is a mutation. This is not a mutation. This is just something that happens. So you do change the regulatory molecules in the cell. This one was a little bit tricky, so I did give you just credit for it. Basically, what is happening is the environment is deciding like what genes are being turned on and off, and those genes or regulatory molecules, right, will determine how they look. It is a little bit tricky, and you won't see this too much. You will see though the whole how genes turning on and off will affect the cell or the differentiation, what cell becomes. As fertilized eggs divide, the cells differentiate because, so again, the cells become different because of our genes turning on or off. That is cell differentiation. So they contain a lipid bilayer, they can clump together, they metabolize sugars, they have specific genes activated. Yes, so cell differentiation has nothing to do with the lipid bilayer. That actually has to do with your cell membrane. They clump together. Cells don't normally clump together. Remember, if they do, that is cancer. And they metabolize sugars rapidly. This is just getting energy. This is respiration. So again, the only one is D. So we have different traits because specific genes are activated. So our cells have specific instructions that are being followed, and that's why they become different. Oh, that is just a copy of the same question. I believe that's it, guys. We already finished reviewing the exam, so again, make sure that you review your notes. Once you are done, you can finish any pending ed sites. You should have a total of 6 quizzes completed. If you brought your backpack and you have your folder, definitely track them. Again, you can always go to your ed site, go under submit it, and then you can get your grade again. I will be checking that on Monday because Tuesday we're on alternate schedule. But thank you so much, guys, for coming to Saturday School. Finish any pending ed site, and if not, you can start watching review videos for any topic that you failed. But have a great Saturday, and I will see you.